Today we have Dr. Cheng and Mason Russo of the Turfgrass and Landscape Pest Management Laboratory at CTAHR on Oahu. So this talk will cover current distribution of hala scale in the Hawaiian Islands, identification tactics, chemical control strategies, dispersal evaluations, forest regenerative impacts, and biological control explorations. So Dr. Cheng is a professor and extension specialist at CTAHR, UH Mānoa. He has 21 years of research experience in biology, ecology, and management of turfgrass and landscape pests. His current and recent research projects in Hawaii include control of coconut rhinoceros beetle, control of ficus stem and leaf gall wasps, lobate black scale on banyans and hibiscus, control of oriental flower beetle, hala scale, which we'll be talking about today, foliar nematode on ornamental plants, and many, many more. Mason Russo is a fourth-year PhD candidate in Dr. Cheng's lab. He spent his time exploring IPM strategies for coconut rhinoceros beetle and hala scale. His research experience includes entomopathogenic nematode and fungal experimentation, systemic insecticide laboratory and field testing, ornamental plant phytotoxicity trials, forest demography, insect surveying, and biological control exploration. He's excited for a future in university extension, industry, government, and conservation. So I'll end this poll. We'll share that. It looks like we have folks joining from Maui, Oahu, and Hawaii Island. So mahalo. And I think I will let Dr. Cheng share his screen and you guys can get started.
Thank you guys for the introduction, and I will just go ahead and share my screen. Share screen, share. Okay, it's loading, yeah. For some reason, maybe the PowerPoint is too big.
Yeah, I think let's give it some time to load.
Yeah, because we have lots of images, photos on the presentation. Yeah. It says not responding, but okay. I think finally loaded.
Okay.
Yeah, can you guys see my slide, the title slide?
Yeah, it looks good.
I'm gonna minimize the top bar, hide floating meeting controls. Okay, all right, I am ready to go. All right, good afternoon, everybody. Welcome to the hala scale talk this afternoon, and we will be covering a lot of multiple aspects of hala scale research in my lab. But we made this title to be very broad. Basically, it is to understand hala scale and develop an IPM program to control it in Hawaii. And this is myself, and people just call me Cheng or Dr. Cheng, and I have my co-author, my PhD student Mason, on this talk as well. And we are part of Department of Plant and Environmental Protection Sciences in CTAHR, UH Mānoa. All right, let's go ahead and get started. Okay, let me see. Let me make sure it goes smoothly. Okay, so first of all, I want to acknowledge my former and current lab members. Not all of them contributed to this hala scale talk, in terms of research or data collection, but we work as a team. That's why I want to acknowledge all my former and current, lab members in the beginning. All right, so the outline, the structure of this seminar would be essentially 5 components. The first component would be the introduction of hala scale and chemical control that we identified. And then we will talk about a statewide survey of hala scale dispersal study, and the potential impact of hala scale on forest regeneration, on hala forest regeneration, apparently. And then the last part would be hala scale biological control exploration. So I will cover the first part and the last part. So the middle 3 part, I will give the floor to Mason. So then basically when I finish this part, I will stop sharing my screen, and the Mason will start sharing his screen. And when he finishes this part, he will stop sharing his screen, and I will again share my screen for the last section of this talk. So for most of you guys, I'm pretty sure most of you guys are familiar with hala scale. So it is a part of the palm scale family, and the insect is only known to infest plant within the pandanus genus. Outside of Hawaii, only these 3 locations are hala scale positive, worldwide. That's what we know at this point. So those are the Thailand. Sorry, I think I have a typo. It should be Thailand, not Thailand. Thailand and Singapore and also Java. Java is apparently a part of Indonesia. But then we later on, when we talk about the exploration, you will see that we actually went to another island where there is a lot of hala trees growing, but we did not find a hala scale. So currently, the distribution of hala scale is still correct based on what we did, 2 months ago, about 2 months ago. Hala scale, they are at the larvae stage. For scale insects, most of the scale insects, they have we can roughly separate them into 2 life stages, the initial, the crawler stage, and also the adult stage. At the crawler stage, they have pretty good mobile capacity. They can move around, but they're also very light. They can be actually spread through infected leaves and the fruits and also wind as well. Mason will talk about the dispersal study, kind of to give a little more details on this perspective. And then what do they do? Heavy infestation like this one, you see, it can eventually kill the young seedlings and potentially prevent forest generation. But on the leaves themselves, usually they start with the underside, the lower, the low underside of the leaves. And then when the infestation is very severe, too many of them, they actually can come on top of the leaf as well. And then they can cause the leaves to discolor, become, from healthy green color into yellowish, discoloration. And also they can stunt the plants, twisting, yellowing, and leaf deformation, and short leaf length, and crown drop, and the other, all you name it, all these potential damages they can cause to the plant health. And in reality, when the infestation is really, really heavy, they can potentially kill hala trees. And in response to that, some of the cases, people have to cut down essentially get rid of the heavily infected hala trees in order to protect the still healthy trees which have no hala scale established on them yet, if they are relatively in close area. And then, but why we care about hala trees and why we care about hala scale, as we, most of you guys already know, I'm pretty sure, hala trees have both economy and cultural value, important cultural values in Hawaii. From my perspective, initially, because my lab is turfgrass and landscape pest management. That's why I began to work on hala scale as a pest back in 2016. We will touch on that a little bit later, a little bit more details. But because hala trees are very popular landscape trees throughout the Hawaiian Islands, that's the initial reason why I began to work on hala scale, because it's kind of a part of my job to work on this pest from the landscape pest management perspective. And then, of course, we have a very big weaving community that in Hawaii that they value the cultural value of hala trees very much. And it is usually when if you ask many weavers, they probably will say, okay, the hala scales will definitely damage the quality reduce the quality of hala leaves, and they apparently will not prefer to use hala leaves infested with hala scales. But then still some weavers might say they can still use it, but it's not apparently preferred because they don't look as green and as healthy anymore if they are heavily infested with hala scale. So initially, this is how I began to work on hala scale. Well, next slide, I think. Just a quick description of where are hala scales right now. In Hawaii, it is not like a new pest. It was first detected in Hana on Maui back on 1995. Right now, we all know that it is widespread on Maui, and limited infestation confirmed on Oahu back in 2013, 2019, and 2020. We will come back to the 2020 infestation a little bit later, and also Molokai on 2014. And right now, it is also widespread on Molokai. We will have a little bit more details when Mason talk about the statewide survey, later this afternoon. For Hawaii Island and for Lanai, there was one instance limited hala scale confirmation, but they were both eradicated back on the Big Island and on Lanai. That was back in 2019. It was never confirmed on Kauai. So Mason will talk about that in detail when we talk about the statewide distribution. And then on Oahu, come back to Oahu. 2013, that was the hala scale. That was year when hala scale was confirmed on Chaminade University campus, which is pictured here. This is where I started to work on hala scale back in 2016. And then 2019, some hala trees were hala scale positive, confirmed in Waikiki Beach Resort area hotels on their landscape trees. And then 2020, hala scale was confirmed in the lower campus of UH Mānoa campus, which is relatively actually close to the Chaminade University campus if you think about it. So back in 2016, Department Ag staff member Derek, who actually, well, he retired, a couple years, several years ago. He's no longer working at Department Ag, but back then he approached me and he said, Dr. Cheng, we have this potential hala scale, trial project. Do you want to set up a trial at Chaminade University campus? And we collaborated and we started working on hala scale project back in 2016. It was not like a formally designed, replicated research plot type of study because as you can see on the map, they don't really have a like big area with even distribution of hala trees and hala scale. There was probably 15-ish hala trees on Chaminade University campus. All of them were infected with hala scale. And then, but we were able to only do some, kind of like a small scale, almost like a demonstration type of, project. So we just, there's no real like a solid scientific type of research. It was simply not feasible to do it. And then, but as you can see, some very heavy hala scale infestation. This is how you tell hala scale. They are very small. They are actually smaller than many other scales insects or mealybugs or whiteflies, which are closely related, to scale insects. But then you see this in many cases, you see a lot of, usually on the underside of the leaves, you see those black dots, and some of them will have this whitish colored fringe, surrounding the black dot. Some may not have it, may not have the white fringe, but then, some do have it. And then those are typical hala scale. But then there are also several other more or less similar, scale insect that can be, attacking the hala, leaves. But then there's some confusion. But it's usually when, say, for me and for Mason, we are experienced with hala scale research, we can tell them, right away. But for general people's eyes, public eyes, they may look, disease because you don't necessarily see them on the top of the leaves, and then they may look yellow. You might think it's a fungal infestation or something like that. But underneath, on the underside of the leaf, you are able to see these black dots, very small, relatively small, and then those are the kind of like the visual confirmation of hala scale. And then back then, we tested simple treatments. We want to kind of just have some ready-to-go treatments available for folks to use if they want to treat hala scale. We tested imidacloprid via injection or soil drench. And then we also tested acid with injection. So it turned out after 6 months of data collection, either injection or drench of imidacloprid was able to have achieved 70% mortality over 6 months post-treatment. So that is a relatively, reasonable like level of mortality. So then we actually right now, if somebody asks me how to treat hala scales, this is still like the legal recommendation I can recommend because when I talk about chemical control in the next few slides, those are new chemicals, are not necessarily labeled for hala scale control legally in Hawaii yet. But imidacloprid is legal to use to control hala scale with a decent efficacy. Then also at the same time, Derek ran another small field trial with hala scale. He was testing dinotephrin, another systemic insecticide. Also, it was via soil drench, was pretty effective too, about also 70 up to 80% mortality after several months. So both of these can be used to control hala scale legally here in Hawaii at this point. But then I got some funding to work on more like a real, really formally designed experiments to control hala scale using newly available, insecticides. This was done back on maybe several years ago now on Maui. This was relatively recent. And then we did this field trial for two repeats, year 1 and year 2, exactly the same treatments because we want to be able to repeat the experiment and see if we are getting similar results in those 2 years. And why we do it on Maui, because we have to fly over there every time and do the data collection, because as I mentioned, on Oahu, hala scale has not been confirmed all over. So it's only 3 locations, 3 spots. sorry. Back in 2020, and then I will come back to that point again. 3 now is 2. But then we do not want to bring hala scale to a place, say our Magoon research station, where there's no hala scale infestation at all. We don't want to do that. And but on Maui, as I mentioned briefly, widespread already. There's no problem. You run the experiment anywhere on Maui. So we collaborated with our colleagues in CTAHR. Actually, we have an extension office there, and we set up the field trial over there. So the rating system that we use is 1 to 5. 1 is means no scale, and 5 means the plant dead due to hala scale infestation. But then when we begin to work on this project during the year 1, like in the very beginning, we realized that the hala scale, although the visual rating is very is commonly accepted. However, for these scale insects, even they are cured by our treatments, they are still going to be attached to the leaves of the the hala leaves. And then from visual, if we only rely on the data collected by visual evaluation, we might see kind of confusing result, because even they are dead, and we still visually evaluate that how many scales are there on the leaves, right? So then I come up with this tapping removal system. So basically, if the hala scales are killed by the treatments, they are still attached to the leaves. However, they are not tightly attached to the leaves anymore, not like the living hala scales, right? Because the living ones, they are able to attach to the leaves very well, very tightly. But the dead ones, they are simply attach them kind of loosely hanging there. But then if we use this tap to tap it, and if we are able to remove the hala scales from the leaves, that means they are dead hala scales still attached on the leaves. So this gives us an indication of actually some of those hala scales are already dead. Then we can kind of rule out that confounding factor. So then my postdoc tested this approach that we developed. It was working actually fairly well, and also more or less matching the direct visual evaluation. Although we were hoping, we were a little bit concerned with when we only rely on the visual evaluation without this tapping method. So then the idea is that the more you can remove by this tapping method, the better, the more effective the treatment was, because they are killed and then they are not hanging onto the the leaves anymore tightly. Then you can actually remove the dead hala scales with the tap very easily, but you cannot do that for the healthy hala scales. So that's the quick idea. And this is the method in action on the year 2 study. And these are all the treatments we tested on Maui, in our formally designed research. We don't have to go through all of this, but the experimental design is like, for this, because we received funding to actually do this in the most proper way, in terms of experimental design. We had replications, we had multiple treatments, and we randomly assigned the treatments across the plant. And then we also had automatic irrigation, for all the plants. This is the protection, the PPE here is good. I mean, it's probably a little bit overkill, but it's always good to have extra protection just in case, right? And then we have all the data collected based on the experimental design, the protocol, and we evaluated the treatments effects on a routine schedule, and then we come up with this data presented here. We have more data to present, but I only present the key infestation level result. The year 1 data was handled by my former postdoc. He finished the year 1 data collection and he went back to his own country, PNG. And then that is when I assigned Mason to this project to start working on the second year project. This is when Mason began to actually work on hala scale back in probably maybe 2 years ago, I would say. So year 1 data was collected by Philip. Year 2 data was collected by Mason. So the good thing is, although we were hoping to see very much the same result, although it usually for field research we might not be able to see exactly the same results. However, we see kind of similar pattern or trend between these 2 years data collection. What we have here is these 2 treatments, Altus and Talus. Both of them were able to reduce the hala scale infestation based on year 1 data and also year 2 data collection. Although there's a little bit of difference. This treatment somehow was effective in year 2, but then not very effective in year 1. Also quite relatively effective as well. But then these 2 treatments really kind of stood for this data collection. And similar pattern is when we look at the data collected from the tap removal approach I mentioned earlier. So basically, you want the bar to be higher. The more you removed, that means the treatment were more effective. Then in this case, again, in year 1, both of these treatments were the most effective treatments, and in year 2, again as well, very high, ranked among the top 3. Although we have another kind of treatment joining the effective ones, but still the pattern hold true that both of these treatments in both years data were able to result in the best treatment among this chemical treatment arrangement. Then this basically was the 2 treatments identified. They were the most effective based on both of the data collected. However, I want to still mention it again here. Both of these treatments were not, these are not ready to be used to control hala scale in Hawaii, because they are not labeled for hala scale control on hala trees in Hawaii yet legally. So then the common, if people ask me the treatment recommendation, it will still be imidacloprid and dinotefuran because they are legal, they can be legally used. But this too, the idea of this research is to now convey the message to the manufacturer of these chemicals and kind of request them to add hala scale and hala trees on their product label and then register that in Hawaii. So then only after that, people can use these treatments to treat hala scales legally here in Hawaii. And there were some, confounding little bit factors. Maybe there were some residual effects between the year 1 and the year 2. But in general, overall, we are comfortable to say that both of these treatments were very effective in terms of hala scale chemical control. And we published the paper last year on one of the ES entomology journals. So that was the chemical control part. I also, this is ongoing right now, ongoing chemical control. Also on Maui, again, the same location, because we don't want to still spread the hala scale on unconfirmed locations on Oahu. And right now, it has been several months since we established the plots, but the seedlings of halas are small at this point. So we are simply waiting, taking care of the plot and waiting the halas to grow bigger, then we will be able to apply the treatments. When they get a little bit taller, then we are able to install a new set of treatments, which are different from the one I showed you before. Then this would be is ongoing, but the applications of treatments have not happened yet. This is coming up. And then remember, when I talk about the Oahu situation, we used to have 3 confirmed locations for hala scale on Oahu. The last spot was UH Mānoa lower campus on Doe Street. So then the landscaping department, who take care of all the trees on UH Mānoa campus, reached out to us, and they were concerned with the hala scale infestation over there. So then we reviewed the situation, and multiple trees were heavily infested with hala scale. So at this point, it's still treatable, but the manager of managing the landscape sites on UH Mānoa campus, she actually proposed to just cut down all these hala trees and get rid of them. I know it's kind of sound like sad, because they try to eradicate the pest by eradicating the host plant. But the way they think is they sacrifice these maybe 10 or 12 hala trees on the lower campus, but then we have a lot more healthy hala trees on our main campus as well. And then at least those trees, they can be more or less protected, not not like infected by the hala scales potentially that can spread over there. So that was the thought for this overall eradication. So right now we can say that on Oahu we only have 2 positive sites for hala scale. Initial one, which is Seminole University campus, and the second part would be the Waikiki Beach resort area. Then hala scale, well, since I have been working on hala scale since 2016, it was never a big deal. But in the recent years, more and more people begin to pay more, have more interest in our research. And we have been interviewed by, like the radio station and the TV, this and that. So then that was kind of like the journey where Mason began to work on hala scales in a much more, intensity type of, research. Okay, I will stop sharing here, and I will ask Mason to, it's essentially the same slide, but Mason will share his screen from here on, yeah.
Thank you for the introduction, Dr. Cheng and Hawaiʻi. So I'm going to share my screen now and we'll continue where we left off with that. Okay, can we, is this visible?
Yeah, it's visible. I can see, but it's not the actual slide. Oh yeah, right now it's good.
Perfect. All right, thank you very much. All right, hello everybody. So we're going to go on to the next few parts of we've determined some chemical control methods for the hala scale, and we want to also take a look at ways that we can figure out its statewide distribution and also forest regeneration capacities and ways it can be dispersed. So we went out to do a comprehensive statewide survey. There's multiple surveys that took place over a few years, where red pins indicated hala scale positive, yellow pins indicated hala scale negative. Now you can see that we when we went to these islands, this is the Wahoo survey. So those populations that Dr. Cheng mentioned previously are included here. You can also see that we did eradicate the Oahu population, so we can have less red pins on the slide when we update this. There is a widespread infestation in Waikiki of hala scale, which is good and bad. So it's good if we ever bring biological control agents to Oahu to rear for the hala scale, we can actually extract source population from Waikiki for rearing. It's bad because it could be a pathway for it to spread throughout Oahu or other islands, hypothetically. So we did a comprehensive survey of Molokai. There's one variegated hala by Pu'ahoku Ranch that does not have hala scale, but is widespread on Molokai. On Lanai, they had a previous infestation by the Four Seasons, which is now confirmed eradicated. And they had eradicated that by, I believe, burying and spraying all the trees that were infested. And then in Hawaii Island, the Pai'ilo infestation was eradicated by cutting down and burying as well. And all the trees nearby were treated with, I believe, imidacloprid. And when we do these surveys, we want to target potential points of entry, such as we want to go to hardware stores where they can be selling plants, areas of mass tourism where there's hiking, where maybe it can spread on backpacks, and also plant nurseries and airports, places that could be potential pathways for the scale insect to spread throughout the through unknown means. And on Kauai, we didn't find any hala scale. No hala scale has been recorded to this date. But we did inspect to the very intense capacity we did everywhere else. So Maui, we talked about previously, how it's known to be widespread. On Kaho'olawe, the only hala plant had died in 2020. Oahu, we have 2 established populations, and then Molokai is widespread, while Lanai and Hawaii Island were eradicated, and Kauai does not have a population, and I don't think Niihau has hala scale. All right, so now we're going to get to the next portion. We determine the statewide distribution of this pest. What are the ecological impacts of this pest in the wild? So if we have extensive hala forests on Molokai, Maui, Hawaii Island, and Kauai, with some minor hala forests on Oahu, we can do a chronosequence or one-time study evaluating the current health status of these forests. This image from Gallagher 2012 shows in the figure B the current pandanus distribution, which shows that Maui County has a lot of hala forests, and also Hawaii Island and Kauai have a lot of hala forests as well that could be potentially vulnerable to this pest, or it could not be. So this is why we set out to do this. So this, I guess the slides are slightly moved here, so we're also gonna quickly interrupt that for the dispersal studies. We wanted to see when it gets somewhere how quick it can spread. So hala trees, we wanted to see when it gets to another island. We set up a trial in Maui to see how does this pest move within a field plot. So at this same field plot we did previous chemical trials on Maui, we grew plants on Oahu that did not have hala scale. We had them inspected prior to shipping to Maui and did not ship them with soil to prevent coconut rhinoceros beetle from being brought over to Maui. And then we had this field plot that was then set up to determine if the hala scale would spread from a center plant in the middle to the outside grid. So in this situation, there's no hala trees nearby, and we did heavy inoculation of the crawler stage on the center tree, and this would be a good field comparison. If the hala scale were to spread to a new airport or port or many other ways of interest, will it spread to nearby areas, and how fast will that process take? Now, this image indicates how fast that process took. So you can see within 3 months, we had populations of those crawlers becoming adults on the center tree. And then within 7 months, it had spread downwind to 2 trees within the plot. And then 8 months in, you can see that it spread sporadically up and downwind from the plot. And then by 10 months in, it was widespread. It was detected on every single tree within this smaller field plot, which shows the nature of this insect and how it can spread. So with this in mind, it was interesting to know from a population biology perspective that there's over 300 adults on the center tree about 1 year into the study, while some of the ones only had a few individual scale insects present. Now, this is not parthenogenic like other scale insects, so there are both male and female scale insects present, so it does take more time for populations to grow, but as you can see, with proper time frame, it will spread throughout the tree. Okay, so now we're back to the forest tomography study. If you remember the previous image of the islands with and without hala scale, we want to go out and how do you quantify a forest on another island without user bias, and also to take a relatively decent ecological snapshot of what is going on. So this was the question I worked on with Dr. Tamara Ticton, where she helped mentor me on how to collect proper unbiased transects, which was through the utilization of a random number generator. So I'd walk to these forests, I'd pull out this random number generator piece of paper, which tells me where to walk at a compass degree angle. I then would follow that angle and set up a rectangular grid that was 20 by 6 meters. And I would put together as many grids as possible within a section of coastal hollow forest. And then once this transect was made, sometimes they're pretty quick to make, sometimes they were challenging depending on the terrain. We then would quantify the demography or the amount of each life stage of trees within each forest. So we catalog trees into EG, 0 to 1 meter, small, 1 to 2 meter, medium, 2 to 3 meter, and large, 3 meters plus. And now you can see below the amount of different forests we were able to survey on each island with the number of transects that were collected within each grid. And to give you some perspective, sometimes one transect can take more than an hour, depending on how challenging the terrain is and how many trees there are. So this was a very comprehensive and physically demanding field project. While we were collecting this demographic data, we collected more ecological factors. We used a spherical densitometer to quantify the canopy coverage at 5, 10, and 15 meter intervals within each 20 meter transect. And we would take 12 measurements per grid forward at each point. We also would quantify the invasive plant understory using this rating system below, 0 to 20, 21 to 40. And we didn't really need anything greater than 40% for this survey. Also, we would do the recruitment ratio. So we talked about the size classes, but we want to talk about recruitment. So that is a reproductive tree by the non-reproductive stage, which can be a representation of successful regrowth within a transect. So we have those stages of the previous size mentioned before, EG divided by LG, S divided by LG, and M divided by LG. And this was the final ecological characteristic that we quantified within these transects. Now, what do these transects look like? This is what one looks like without adult trees, where you climb up a tree and take a picture of it. So you can see here, it goes way off into the end, and this has some probably some small to medium halas present that would be in this grid. And you can see when the random number generator tells you to walk in a specific direction, if you look carefully in this image, you will see measuring tape. And I walked through some of the most challenging terrain I've ever experienced, and to maintain these perfect 20 by 6 meter grids. And even though you don't want to do it, and there could be user bias this is why you have the random number generator, because you have to follow these very difficult lines within safety parameters, of course. So this is how we were able to quantify so much land, even if the terrain was very challenging. So on Maui, we surveyed the Hana area. I see Mike is here in the participants, so why not on NTPG. Thank you for access to the many landowners throughout these next few slides and permits and everything we had to obtain. We could not have done it without you. So this is some images from Maui. Molokai, we surveyed Kilkipua Peninsula, Hakaanu, Kahoa Lake, Wai'ehu, and Halava Valley. Now, we're going to see Kahoa Lake here. I want to show for the trained plant eye here, as the helicopter is going down, you can see the yellow necrosis of a heavy hala scale infestation, which shows that even in the most remote areas of Molokai, where there is extensive remote hala forest, there also is widespread hala scale. So right here, you can see as we're about to land, if you look over here at the canopy of these trees, you see that yellow necrosis Dr. Cheng described, showing what happens when you have heavy infestations. This was thanks to coordination with MOPEP and their excellent scientists, specifically Anne and her crew were amazing for getting us out there. So on Hawaii Island, we had coordination for permits for Puna, Hilo, and Waimanu. And you can see here, this is a greener tree, so it shows a healthier indicator without hala scale. And Kauai, Nainakai, Princeville, and Haena. All right, so these next few slides don't have error bars because when you run a general linear mixed model or multiple mixed model analysis, it shows there was statistical significance by location. So to avoid any bias indicated by location, we're just going to show you the raw data that's indicative from all of the data we collected, and then we're going to go into the statistical analysis. So for overall canopy using the spherical densiometer, we can see that Maui had the least canopy coverage of any island. In terms of the understory was below 20% for invasive plants, so that was a negligent characteristic, but still was something that was important to collect at those 5-meter intervals. And then for the mean elevation, Kauai had the highest elevation per transects on all the islands. And finally, here's the total count of hala trees on each island, and you can see that in terms of these stages for recruitment and reproduction, you could take a general look, and you would want the middle 2 bars, which are red and then green respectively, would be indicative of successful regrowth. And you can see that there might be less of the green stage on Maui and Molokai, which are indicative potentially of a future problem. Now, when we get into the actual recruitment ratios for this, we did an analysis of every single island. So you can see within each island doing the recruitment ratio of these life stages. And there was no statistical significance with this study. But once again, I want you to look at the trends that we're seeing here, which there is less of the green M stage, which is the last stage before what would be the trees where we see the most flowering or reproductive structures. So that is a sign for concern where we're not seeing, within the recruitment ratio, we're not seeing that M stage present within these transects. And then when you compile the data into a general assessment of is there hala scale, yes or no? No meaning Kauai or Hawaii Island, yes meaning Maui or Molokai, and you can see that this trend is amplified once you do this. Now, after all that walking, I'm glad that there was some statistical significance. So running a multiple regression model with more predictors. The previous one was only predictor hala scale yes or no or by island. When you incorporate the ecological factors we collected of canopy coverage, invasive understory, elevation, and hala scale yes or no grouped into this analysis, still with location as a random factor, due to that statistical significance, you see that islands that have the hala scale have less regeneration of the SLG recruitment ratio. So that is statistically significant. You also see, if you look at this image here, a negative correlation between canopy and small trees on islands without hala scale. Now, I don't want to lose you all here, so let's think about what that means. So when the canopy, remember the spherical densiometer where you see the full forest cover and everything that's going on with the canopy. So you would anticipate when there's a full canopy, there is less trees of different size classes because they're shading it out or there's leaf litter preventing that regrowth. Now, on islands with without the hala scale, as that canopy decreases or as these older trees are dying, you are seeing an increase of that recruitment ratio of the S size class happening within these areas, which is indicative of successful regrowth. So that's a trend you will see. But then when you do that same analysis for islands with the hala scale, Maui and Molokai, you are not seeing that trend, suggesting that the SLG recruitment ratio is negative. There's a negative correlation, and also maybe less overall future recruitment of hala forests. So what all that means is these forests on your right looking lethargic compared to the ones on your left, and there may be some recruitment issues in the future, decades or even maybe a generation from now, if this continues to be a widespread pest within these forests. So summarizing this, there's less canopy coverage on Maui, with the invasive understory being relatively negligent, and with the SLG recruitment ratio that I mentioned, it can be partially attributed to ungulate grazing, but that wasn't in every single transect, and we did have the randomization by transect to account for that data variability. So this suggests that this may be a recent event now that the scale has been present for 15, 20 years in some of these sites. So if this was impacting, it's not impacting the M stage, which is the stage before large, but because it's impacting the stage below that, it may indicate that we're not going to have successful recruitment of trees in the future. It is also possible, as I said, that it does decrease the regrowth, but right now it's not having immediate forest regeneration where there's no regrowth. Right now it may just be slightly less, which may be a trend that will continue. But the SLG decreased ratio is concerning for long-term regeneration capacity. But I think primarily we need to focus on ungulate control, and also some, as you can see here, the ungulates on Molokai have ate every single, there's some endangered plants that used to be here, and everything has been grazed on this one peninsula off of Molokai. So the deer and goats need to be managed here. And yeah, so anyway, there were limitations with this. I couldn't walk across every hala forest in the Hawaiian Islands, but I did as much as physically possible. There were kapu areas that I definitely did not want to go into and was not appropriate. There was private property that also I couldn't get into. Hala forests have very steep gradients where they grow. So it's not very safe to collect proper transects on these cliffs. I couldn't get permits with Kalapapa National Historic Park, and there's one forest peninsula off of Waimanu Valley that I was unable to access. You can see that steep gradient here. There's a hala forest all the way down the mountain in Waimanu Valley, but it's not physically possible on that gradient to collect the data. Finally, this image here is something I really want you all to think about. So if you see this ironwood forest slowly approaching the hala forest, this is something I encountered on Maui, Hawaii Island, and Kauai, where even though the hala scale is a very bad pest for hala forests, I think ironwood, because it changes soil chemistry, is pushing out hala trees throughout its entire natural range. And there are some manuscripts indicating this is happening in other places in the world, but right now it's not considered a pest species. So that's something that you could think about in the future when you're managing properties that have ironwood, maybe to cut them off at a line where they won't encroach on any native forest. Otherwise, I've already mentioned development and ungulate control. And finally, I want to talk about just before we transition to Dr. Cheng, who's going to finish with the biological control portion of the study. This is a picture of Pandanus vandermeerschii from the HANA NTPG, which is resilient to the hala scale, which shows there's Pandanus varieties that can be directly next to a Pandanus tree that is infected with hala scale that the scale insect will not target. So I'm going to pass this on to Dr. Cheng, where he's now going to present the biological control survey we just completed, and then he's going to conclude the seminar.
Okay, thank you, Mason. I'm gonna share my screen again. All right. Okay, this is the one, I think. Okay, all right. So this is the last part of our seminar. It's been, oh, I can't believe it's already 3:47. So essentially, I applied to a USDA grant and then was funded to a part of this project is to actually to test a new chemical control on hala, hala scales, which is the one ongoing on Maui. I showed you. The other part of this grant was to actually to look for potential biological control agents in other locations where hala scale is positive, is confirmed. As I mentioned in the early this afternoon, right now, except Hawaii, we only know that Singapore, Thailand, and Java, one island in Indonesia has confirmed hala scale infestation. So then we wrote these 3 countries specifically in our proposal to look for the potential biocontrol over there. This is a map where we show where we surveyed, and again the red means positive hala scales. But then we will have a little bit more detailed discussion here. And okay, I'm going to again do this remove this hide floating. Okay, now I can see the whole screen. All right, so in Thailand, we surveyed, well, we had about a little bit over 2 weeks, for this entire trip to hit 3 countries. So each country, we had essentially 4 days, and we can only go, so many places within 4 days, right? So in Thailand, we went to 2 main areas, Bangkok area and also Phuket Island. So both places have, quite some hala scale, hala trees, especially the Phuket Island. But then we did not find a hala scale in Bangkok area or the Phuket Island. However, there's a kind of like a very interesting side product that come out of this Bangkok visit was my colleague there, my contact there. So she has been actually working with biocontrol for CRB over there in her, department, in her, facility. And this is, we most likely, I will collaborate with her in the future for CRB control using biological control agents as well. That's another direction that, we are going for the CRB control as well. And let's see. Okay, so Phuket Island, as I mentioned, we did not find any hala scale there. Then we went to Indonesia. The first stop, the first country we went to was Thailand, and then from there we went to Indonesia. We went first to Bali Island, which is the island where hala scale has not been confirmed, but it's right next to Java Island where hala scale is confirmed. So we were expecting, or we were hoping to see hala scale there. And then when there is a hala scale, there's a potentially hala scale biocontrol agents. But we did a pretty thorough search on this island, but then we did not find the hala scale. So that basically the current worldwide distribution of hala scale still holds true. That only includes Hawaii and Java Island, Thailand, and Indonesia. So then back in Jakarta area, the metro area, and then where we found hala scales, mostly in part of this botanical garden where we found the hala scales, and there are several, multiple hala species there. Hala trees are not like quite as common landscape trees in those countries like we have in Hawaii, but in this botanical garden, there's a clustered hala scale hala collection over there. There are multiple hala species, similar but different. As you can see, we found the hala scale infested on some of the species, but not on some other species, kind of like echoing what Mason mentioned, some species of hala trees, hala species are actually resistant to hala scale infestation. And then also we found two lady beetle species that we saw them actually feeding on hala scales over there. But then the lady beetles are generally considered as generalists. They are not specialized, biocontrol agents. And one of the species we pretty sure we have it in Hawaii here. So then, and also again that we do not have the permit to bring anything back to Hawaii. Plus, the one of the lady beetle species, the bigger black one, are actually we are pretty sure we saw it in Hawaii here. So then we basically left the specimen with my collaborator over there and pending their further identification to the species level, or at least to the genus level. And then we move on to Singapore. Singapore is many places are fairly heavily infested with hala scale that we collect, we were able to see in hala scale, situations. And these are the two the hala species, pandana species that are susceptible to hala scale. This is the lady beetle that I'm talking about. And some of you probably realize that, yes, we do have this similar, at least similar. Superficially, it looks similar. Very likely they are the same species. These are the the not your typical lady beetle with black and orange color, but they are pretty much only black, but very small compared to a typical lady beetle species. And then we saw them feeding on hala scales. And again, we were not able to bring anything back because we do not have the permit. But then we left these two lady beetle species, very similar to what we were able to collect in Indonesia. One bigger one and one smaller one with my contact over there in Singapore. So then they will be able to further identify those lady beetle species to the species level. So that's what we come up with, all with this big effort of 2 weeks exploration trip to these 3 countries. And then most likely what we found, we feel that the hala scales are not, those places are probably not the native range of hala scale, because many of them are not being used for very commonly. But then we do have a lot of hala trees in forest in some of those islands as well. But then we don't, the hala scales are really found in urban areas where those ornamental or landscape hala trees were brought in to some extent. So then we were not sure whether those the 3 countries are actually indeed the native range of hala scale. So that's kind of remains more or less a mystery. So what's next? So the next is apparently we left the 2 lady beetle species in both Indonesia and Singapore, and our collaborators over there will hopefully be able to identify them to the species level, or at least to the genus level, so that we can report, we can include that in the project report. And as I mentioned, these introductions may not be the native, hala scale range in those 3 countries. But then also there are other places where the hala trees are endemic. So then those, it's likely that hala scales might be there, but again, there has been no confirmation or report from other places. For example, I think Madagascar, Sri Lanka, and Taiwan. We can only say potentially, but there's no literature to confirm there's actually hala scale in those countries. But there are plenty of hala trees in those countries. So it's a likelihood, but it's not a confirmed situation. Yeah, that's all we have. And I always have this disclaimer if my talk has any chemical control or included in my talk, then because I do not want people to think I'm promoting certain chemistries, chemicals. This is my standard disclaimer page. My lab page with my lab website and also acknowledgement for the funding, the collaborations, and the people who, when we go to Singapore and Thailand and Indonesia, it's like totally friend, strange countries for us. We had it was actually very good that I was able to find collaborators in all of these places, and they at least help us quite a bit. So we want to acknowledge those, and also for all the survey work that the folks on all the neighbor islands who helped Mason with all his survey research and other things. Yeah, that's all we have. And the last page, we see some happy faces when Mason was in action doing the survey work mostly. And any questions or comments? Let's see the time. Let's see. I'm gonna stop sharing for now. And 3:56. I had a project. I had a funding before and made a hala scale kind of video to promote the awareness of hala scale in Hawaii. And then, but we don't really have the time to playing that video. That video is about 5 minutes long. Yeah. But I will stop here, and we can take questions if there's any. Yeah.
Thank you, Dr. Chen and Mason. That was a lot of great information to be shared around. I learned a ton in your talks. I just wanted to get over and check out our chat real quick to see what the questions are like. I did just wanted to ask you, Dr. Chen, if we can get a link to that video that you had mentioned.
Yeah, I have not uploaded the link to my website, but I can essentially email provide the link, the video to you guys upon request. Yeah.
Perfect. And we can share it with our folks. Awesome. So I think we got a couple questions in the chat. Let's see. So from Mike Obgennorth, is Bali really a good place to look for hala scale? It's very urban. Was there extant hala forest there? It doesn't surprise me no scale was found there.
It was actually less urban compared to, say, Jakarta area. Bali is a very popular, I guess, place for, many people go there, but there are extensive hala scale, forests, not quite as many as, say, Phuket Island, but there's plenty hala trees over there, and it's right next to Java Island. That's why, that's the only really, in initial thought, there's no reason to go to a place where hala scale is not confirmed, because we are not, not only looking for hala scale, but really more importantly, we're looking for the biocontrol agents of hala scale. But then Mason did some preliminary kind of arrangement search, and we believe it's worthwhile to take a trip over there because there's plenty hala plants over there. And then it's right next to Java Island where hala scale is positive. So then, but it turned out to be hala scale negative still in this case. Yeah.
Right. Good to check it out. Okay. And then one more from Mike. Even with the identification of the Coccinellidae, are there any ladybugs that are known to not be generalists? If they're all generalists, why would we investigate further?
Right, most lady beetles are considered generalists. That's why we are not. If you kind of get the hint from what we summarized, we are not going to invest too much on further investigation into these two lady beetle species, because at least one species we think we have. If it's not the same, but we definitely have a similar lady beetle species, the black small ones, not the the typical one, colorful one, but they are feeding. Mason has seen it, and I have seen it also in Hawaii, on Oahu. They are feeding hala scales. So that's if it's even it's not exactly the same species, but they are doing the same job. For Hawaii, there's no reason, because we are all very much concerned with bringing anything new to the island, in a way, although even they're mostly beneficial. Well, that could be beneficial, but then, we don't know really the long-term side effect of those. So then if that's the case, yeah, we are not going to be very strongly pursue those two lady beetle species at this point, but at least it's good to have the species identified. So to some point, yeah. Mason, if you want to add anything, yeah.
Thank you, Dr. Cheng. Yeah, I wanted to add, so on this trip, the previous literature had indicated these places were positive or even the native range for the hala scale. But upon arrival, we saw similar densities to what we're seeing in the Hawaiian Islands where there were infestations. So there's an interesting manuscript published that shows how when you go to botanical gardens that have a collection of the same genus, if you source the locations of the collection, you can actually find where a pest was brought in. So we analyzed the pandanus trees from the collections on Singapore and Indonesia, which we got a bunch of different species names of pandanus. So from where we went, I don't think it was anywhere within the actual true native range, but that is where the hunt could continue. or that I think that left Taiwan, Sri Lanka, and Madagascar, with both Indonesia and Singapore's collections having pandanus from Madagascar that were positive. So maybe it was through ornamental transfer, but we can't say for sure the native range still. So hopefully we're going to start emailing some scientists in those countries with the pest alert and to see if they can figure out if it's there, and if it is there, if there's any natural biocontrol that we can utilize.
The investigation continues. Okay, I think we got a couple more. So one from Meredith Buck. Is there, this is maybe outside your scope of study, but I was wondering if you're aware of any research around the use of fire to control hala scale.
So which part is from the chat, right? So
She's just wondering if there's any kind of research around the use of fire to control hala scale, or is the best method just that cutting and burying? Yes.
use of fire. Did you say fire?
Fire, yeah.
Oh, well, we never thought about that way. But let me just say that chemical control is an option. Still, we have the available chemical control there, but it's for small scale, high value landscape plants, right? Apparently not for the forest setting. But it is nonetheless, it is their option. But we never thought about fire. But then the eradication that happened in several places, like on Lanai, on Hawaii Island, also on lower campus of UH Mānoa, they were all like physically cutting down the infested trees, essentially, kind of burn the infested trees, which are cut down, and also soil drench with the chemical that we recommended to make sure, rule out any continuing possibility of hala scale infestation from that local spot. Yeah. If you want, feel free to add anything, Mason, if you want. Yeah.
Oh, that was it. I mean, I think once they're in a contained area, so these were incinerated in boxed units, that eliminates any chance of fire sending crawler stages or other aspects of hala scale's life history into the environment. So I think fire could be utilized if you have it contained, but that was why burying was utilized in the other eradication instances.
Gotcha. Thank you. Okay, we have one from Chuck Chimera. Is there any evidence of resistance to hala scale in any local pandanus genotypes? Like what was observed in the cultivated pandanus shown in your slides? Could other members of the Pandanaceae be susceptible to hala scale, e.g. Freycinetia, arborea, or EAEA?
I think in one of the Mason slide, he did show that one species of pandanus was resistant in the garden, right, in the National Tropical Botanic Garden. And other members, we are not sure, not familiar with that particular, species that Chuck mentioned. But Mason, if you want to add anything, yeah.
Yeah, of course. So within I think we have 4 or 5 resistant varieties. One is on Maui, and 3 or 4 were identified in Thailand and Indonesia and Singapore. And also Freycinetia, I have not seen infestations on those on islands with hala scale. So as of right now, that's a good sign to see. Within the close lineages of Pandanaceae, we're seeing pandanus specific, and typically a pandanus tictorius with some variation. But I think in Indonesia they want to do a test of the nutritions of the pandanus collection to see what factors within the pandanus in their collection are susceptible or not susceptible to hala scales. And so we're those 3 or 4 other varieties with no hala scale.
Hey, thank you. Okay, we have another one from JD Herndon. Could sampling watersheds using eDNA techniques detect presence or absence in remote, hard to reach places?
eDNA, well, it's a nice technology, but basically the resolution is hard to to be 100% sure. Whether there is for example one seed, one hala scale specimen in the water. I don't think it can have that high resolution that be able to detect that. But it's it's possible. But we have not started that way in that direction. Yeah.
And one more from Mike. At the National Tropical Botanical Garden, we have not seen any of the EAEA getting a hala scale, even with so many infected hala trees in proximity. I'm sorry, so that's just a comment to the previous question.
Yeah. So, right. Yeah. Okay.
Awesome. I had like one real quick question. Is there anything that you guys have noticed that puts the hala trees more at risk for this scale? Like, is there like a drought factor, like pruning, like unclean tools, or like environmental setting? Like, is there anything that you guys are noticing that's kind of connected to the infestation of hala scale?
I think it's most likely physical distribution of spread of hala scales. But then the scale insect do have their capacity to actually move around at the crawler stage, just like Mason was doing the dispersal study, right? So it takes time, but then they can be spread in a much faster way by animals, by birds, and by even human traffic. So that's always a concern. It's not only the case for hala scale, it's the case for potentially many other insects and even weeds, seeds as well. It's just common situation. Yeah. Mm-hmm.
I would like to add, with that dispersal study, it's a very slow process at first. So some of the outside trees in the field plot that were infested, without a trained eye, you wouldn't even know there was hala scale there. So I think if hala scale were to spread to other islands, people may not notice for maybe even a decade until like it really takes up in population. So I think monitoring of pathways of distribution pandanus material should be a priority to prevent it spreading from Kauai or Hawaii Island.
Yeah, it's always true to try to prevent them from going to the neighbor islands. We did a similar survey study years ago for the low-bay glass scale survey. And, that's the first time we found actually the low-bay glass scale in garden centers of a big box store on the big island on Kauai. It was the first incidence of hala scale confirmed, actually spotted on neighbor islands except Oahu. Yeah, so it's similar. They can ship the nursery plants, potted plants from an infected area to their islands. That kind of, I don't know whether that's the case for Chaminade University. Bob has a question on whether how it got to Chaminade. We will never know, probably. And we can probably ask them, where did they get those hala trees to plant over there in the first place, right? Then we can potentially say, okay, if they somehow got those plants from Maui or whatever, and then that might be how they get there. But without detailed information, we cannot say for sure. Yeah.
Well, thank you for sticking in and answering all those questions. Those are all great. Let me just check Facebook really quick. I don't see any additional questions. So over here, if you want to wrap up.
Yeah, I just wanted to mahalo Dr. Cheng and Mason for being here today, and also to plug the Hala Festival, which is happening next Saturday at the Maku'u Market from 10 a.m. to 2 p.m. And, invasive species like the hala scale and the coconut rhinoceros beetle is one of the main reasons why we want to have this event, because it really takes all of us when it comes to catching these invasive pests early. So we dropped, I think, does that URL work? We dropped the URL in the chat. We have a bunch of different workshops, talk story sessions. So if you're in the area, please come down. We're super excited. And mahalo to everyone for staying. I know we're running a little late, but yeah. We have one more question from Meredith.
Yeah, I met with Lola Spencer, who's a weaver. Her question was about weavers taking access to prevent a pathway for spreading it. And Lola Spencer, a Molokai, she took me to her grove and showed me a book of hala scale over time. She kept track of when the first got to Molokai, I think in 2007, and her grove at least. And typically, I think now they're telling weavers to freeze material on Maui and Molokai and Oahu before bringing it to other islands for festival. So those dried up leaves can still have crawler stages present if above there is infested trees. So even though there's no more plant sap for the hala insect to feed on in that leaf that's going to be dried and used for weaving, freezing it will kill the hala scale 100%. So that's the best thing I think that can be taken for if you're gonna. She still makes amazing material from her grove, and it's excellent weaving. So I think in terms of how long to freeze it for, Dr. Cheng, what do you think? 2 days?
I would. I don't think I even need 2 days, but definitely, the longer the better. But I relate to this. I think it used to be the case that when there was a question from UH landscaping department who manage all the trees, right? And there was a request for a weaver, I guess, who will be teaching a weaving class on UH Mānoa campus. So then they were asking the permission whether they can bring weaving materials of hala, to the campus, and then that was right around the time where they want to eradicate the hala scale from UH Mānoa campus. So it was kind of like right on the spot. And then our recommendation is, if you are 100% sure your hala materials are scale free, you can do that. If you are not sure, please, just try to utilize the whatever materials on campus to do it, to teach the class. So that was the recommendation. It's kind of related to this. Yeah, but it's really great that the people are aware of this pest, and then they can, that way, people have this in mind to try to slow down the spread of this pest from going to new spots, right? So that's really important. Public outreach, public education of all this. This is part of the purpose of this webinar series as well, I think. Yeah. Mm-hmm.
Mahalo for all of your hard work and research over the years. I know it takes a lot to make this work possible. So we're really happy that you took the time out of your busy day to talk story with us. So mahalo. And again, this webinar is being recorded and it can be accessed from the Hawaii Invasive Species Council YouTube page.
Okay, great. Yeah, thank you for having us. Yeah.
Of course. And I will go ahead and stop the live stream. Let's see.